Okay, so we've gone through all 5 steps of the scientific method. Let's review what those 5 steps are, okay? What's step 1? Ask a question or observe something, right? We've got to observe something that piques our interest, that leads us to a question. Step 2 is what? Okay, now you're going to make a prediction, form the hypothesis. Step 3 is to do what? Design the experiment. So now all 3 of these have been prep phases, right? We had to think about what we wanted to ask, and we asked it, right? We asked our question. Then we decided what did we think was going to happen. That's our hypothesis. And then we designed the controlled experiment, okay? Remind me, what does it mean to be a controlled experiment? Yeah, Nora. Perfect. Only one variable changes at a time, okay? If we change multiple variables at a time, then we aren't sure what is causing the result change, right? So controlled experiment means one variable changes at a time. Once we've got all of that prep work done, we move into step 4, which is really a big step, right? What happens in step 4? Yeah.
Recording and analyzing results.
We record and we analyze our results. And so that means we do the experiment, right? We do the test, we write down our results, and then we draw our conclusion. So once our conclusion has been drawn, right, once we've decided like what we think is true in our experiment, we form what's called a theory, okay? We're allowed to form a theory after our hypothesis has been proven multiple times, right? Multiple times. We've got to support our theory with evidence, okay? So running an experiment one time is not enough to then form a theory about that outcome, okay? We have to test our hypothesis multiple times and have a lot of evidence to support that, okay? So theories aren't just one and done, right? I decided kind of like, okay, then I did it one time, and so I think it's true, and that's what it is, right? We've got to test it multiple times. So a theory is a pretty broad and comprehensive statement about what we think is true, right? What is thought to be true. And it's a broad and kind of complete statement about what we think is true based on our evidence from the experiment. Okay? You see this word several times brought up here in our, right? It's got to be evidence-based, right? We can't form theories just based on what we think. It has to be proven. It's got to be evidence-based. So that's really, really important. All right, we're going to look at a practice problem here that's very similar to one we discussed in class, right? It's very similar to the one I kind of gave you an example about fertilizer and all that stuff. But let's read through this problem, and then we're going to walk through the whole method just with this example, okay? And we're going to see how it comes out. So you want to determine the effect of a certain fertilizer on the growth of orchids grown in a greenhouse. Materials that are available to you include a greenhouse, orchid plants, water, fertilizer, and soil. You want to know if the orchids will grow best with a weak concentration of fertilizer, a medium concentration, or a high concentration. How will you design an experiment to test different concentrations of the fertilizer? Okay, so we've asked the question. Now your job right now is to write a hypothesis. You go ahead and write a hypothesis in that box that I gave to you, okay? If I apply no fertilizer, orchids will grow best. If I apply low concentration of fertilizer, orchids will grow best. Whatever you think is going to be the outcome is what your hypothesis is going to be. So you go ahead and write whatever hypothesis you want. So all of the class will have several different options here, and then we'll see at the end kind of how that works out, okay? You could also write it in a predict format, right? I predict that low concentrations will yield the best results, or I predict. You could also do it in an if-then statement as well, okay? We need to design a controlled experiment. So let's think about how we can go about doing that. What's the one variable that has to change in my controlled experiment? Yeah, Owen. Okay, that's what we're measuring, but what's the thing that I'm going to control and the thing that I'm going to change? How much fertilizer goes on, okay? So if I'm going to set up a controlled experiment and I've got 3 different levels of fertilizer to test, I need 4 groups, right? Why do I need 4 groups? Why do I need 4 groups? If I'm testing 3 different levels, yeah, why do I need 4, Jason?
To determine, like, also which one, like, without concentration.
Bingo. Good. Which one, the one that has no fertilizer applied to it, what's that going to be called? The control. Good. The control group. Okay, so I'm going to set up 4 groups. 25 of them will receive just plain water. 25 will receive weak concentration. 25 will receive medium concentration. And 25 will receive a high concentration. Right, so we're splitting them evenly into 4 groups. Okay? We're going to make sure that plants are watered daily. And then over a period of a month, we'll be measuring those plants to see which one grew the tallest. That's going to be how we get our data here, okay? As we measure these plants, is that going to be a quantitative set of data or a qualitative set of data? Quantitative, right? Quantitative is quantities, right? It's got numbers that go with it. So it would be quantitative data here that we're collecting. All right, which one of these is going to fit into the control group? Group 1, 2, 3, or 4? Which set of those plants are part of the control group? Group 1. Okay. The control group is group 1. It's the one that receives no fertilizer. Okay, you could also write it as, group 1, right? No fertilizer. And then in the experimental group, will there be more than one of those groups of plants? Yeah, right? In the experimental group, it will contain groups 2, 3, and 4, right? The 75 other plants that are receiving varied amounts of fertilizer. Okay, any questions about how that's split up? Control group and experimental group as a whole don't have to have equal parts, right? We've got 3 subgroups that are within that experimental group. So all of our subgroups are equal, but. All right, in a controlled experiment, all variables must be kept constant except the one variable that's being changed. So let's start making a list of the things that need to be kept constant. Okay, what variables do we need to keep constant in the experiment? Yeah, Grace. The amount of sunlight, good. Did I leave you a space on your slide to write in these lists? Okay, the amount of sunlight. Yeah, what else, Olivia? Type of plant, good. What else? Amount of water, Good. The soil type. What else? Can they be in differing sizes of containers? Nope. So the container that they're planted in needs to be the same. The time or frequency of watering needs to be the same. Right? We can't water one in the evening and one in the morning. Okay, we've got to do them consistently and at the same time. Let's see, what else? How about the location? Right, that kind of falls under the sunlight, but we've got to keep our location the same. We can't grow some in the greenhouse and some outside the greenhouse, right? That's a huge change in the environment of those plants. So we've got to make sure that the location of those groups remain the same. Okay. Any other things we can think of here that need to be on our constants list? Okay, I think we got pretty much everything there. Experiment. Yeah, Nora. Amount of fertilizer. Good. The amount of fertilizer. And what variable is that called? The one that's being changed. The one that we are choosing to manipulate. What's that one called? The independent variable. The independent variable. Okay, so the fertilizer is the thing that's being changed. And that's called the independent variable. We ran our experiment, and now we've got our results. Okay? Group 1, or the control group, grew to an average height of 15 centimeters. Group 2 with the weak concentration grew to an average of 35 centimeters. Group 3 grew to an average of 28 centimeters. And Group 4 grew to an average of 10 centimeters. Okay? So look at your hypothesis that you wrote. Is your hypothesis supported or not supported by these results? Okay? And then write that. Based on the hypothesis that I put up, right, I said that medium concentration would grow best, and that's not the case, right? Which one of our groups really had the best growth?
Weak concentration.
A weak concentration. So in my case, my hypothesis was not supported. Okay, and then you need to write whatever it was with your hypothesis. Okay, how many of you did guess that low concentration would be best? Okay, good, nice, Grace. Okay, did most people say medium? Yeah, okay, good. So at high or medium levels of fertilizer application, we saw inhibited growth, right? And so our conclusion here is that orchids grow best at low concentrations. Okay, is this one experiment enough to form a theory about it? Probably not, right? We would have to do this multiple times. And if we're studying orchids particularly, right, we might use different types of orchids, but that might be the control that we're changing, right? So if we're going to change fertilizers, that would be one thing we have to change at a time, right? We could change brands of fertilizers. That would be one experiment. We could change types of orchid. That would be one experiment, right? But we've got to make sure that if we're going to test just this particular type of fertilizer on this particular type of orchids, we would just do the same experiment over and over again. Okay? If you're trying to make a broad theory about orchids in general, then you could start mixing up your experiments a little bit. Okay? But a theory can't be formed until we've got a lot of data to back that up. Okay? All right, I'm going to go ahead and stop right there for today.